this. So I'll say, let me record it again and post it again. That today is March 1st, isn't it? Mm-hmm. March 1st. We know OSTs are in April, so we have about a month and a half. And we still have 2 units to complete, so we don't have weeks to do this. We have to finish the hybrid crosses now and then start talking about population genetics, which is the next unit, then evolution, then ecology. So let's be clear, we still have to finish dihybrid crosses. We have to monohybrid crosses, all the other things that were posted, all the answers were given to you in class. So if you're in class or you watch the videos, you should have all the answers to everything. So we should finish dihybrid crosses today, if not Monday. We have to do, how do we calculate probability? We have the vocabulary words on the board. All the vocabulary words, we have to change the date, or the date's changed already. We have to talk about, you should know all those vocabulary words. They're all in the textbook. If you need to read something, the chapter reading in the textbook is available to you 24/7. So we have to learn how to calculate probability. We learned already how to do a monohybrid cross. Again, that video is posted. But we have to really make sure we understand how to do a dihybrid cross and what it means to do a dihybrid cross. So I'm going to give you some definitions. I also will post a professionally made video that goes over some of this stuff. I say professional because I'm an amateur at doing this. So when we talked about a monohybrid cross, remember, and I drew it out, we were talking about a single gene, an A gene. And every gene, you have 2 copies. One came from mom and one came from dad. If one person, parent 1, got a big A from their mom and a little a from their dad, or however they inherited that, they are heterozygous. That's another vocabulary word that's on the board. It's been on. Heterozygous means different. So you have a big A and a little a. Now, if parent 1 is heterozygous, maybe parent 2 could be heterozygous. They could have they could have inherited a big A and a little a from their mom and dad. Or they could have inherited 2 little A's, or they could have inherited 2 big A's. We just don't know. But in this case, let's say they do that. We call this homozygous. And specifically homozygous recessive. Again, vocabulary on the board. Recessive means you need 2 copies to see the trait. Dominant means you need to have only 1 copy to see the trait. Again, I'm just going to put the words out here. Dominant, recessive. Gee, if there was only a way we could take all this detail and reduce it to kind of main ideas and vocabulary words so we know what to study. Oh yeah, there is. It's called Cornell Notes, isn't it? So we can't, that was sarcastic. I apologize. It's been a long day. So dominant, recessive, heterozygous, homozygous. All that is on the board. All that is related to meiosis, to how we inherit genes, to how chromosomes are organized. Okay, so if we're going to cross these two and we want to know what's the probability of having a parent have a dominant or recessive gene or what have you, then we would do a Punnett square with a single gene. I think everybody knows how to do that. We would take the gamete, possible gamete. So mom is big A, little a, and dad is little a, little a. We're going to cross mom and dad. It could be plants or bees or birds or humans. It doesn't matter. You're crossing these two individuals. They have this genotype. Oh, there's another word, genotype. Again, all these vocabulary words have been defined in class. Take the headphones off your head. They've all been defined in class. They've all been put on the board for many days now. So if you're going to cross these two, this parent has to make sperm. This parent has to make an egg. This parent has to make sperm. In the case of a human, in the case of a plant, this has to make pollen. This has to make an egg. Whatever their gametes are. Oh, another word. Notice that all the words are on the board. Those are words we wrote as we worked. Gamete. It's another word for sex cells. So if this person is going to make a sex cell, we went through meiosis. We went through it in class. The notes have been posted. You've done work on it. It was due. It should have been turned in. Then you know this person is going to make, first thing in the cell cycle is make copies of each. So you have this one, you have this one. This one came from mom. This one came from their dad. You're going to go ahead and copy them. So you're going to have one big A here. You're going to have another one with a big A here. So there's copies of these. And then this one's going to be little a and this one little a. And then they're going to form a tetrad. And we talked about this tetrad being formed, that this tetrad is going to have crossing over. We talked about all that. So a lot, if you haven't been paying attention, then you're kind of freaking out a little bit now. Crossing over really is not that big a deal for a single gene when you're talking about a single gene, but as you'll see in a minute when we do two genes, all of a sudden it makes a big difference. Where before the two little a's were on, the little a's were copied and they were together. Now you see that the homologs, these are called, another vocabulary word. Look at all these vocabulary words coming. Index cards, do you have them made? Have you been reciting? Have you been reviewing? I don't know. Y'all tell me you have. You turn in your work. Are you copying? I don't know. But you're turning in your work. Those of you that are. Homologs. What is a homolog? It's two chromosomes that have the same genes. What are sister chromatids? Another vocabulary word. Chromosome, the two identical copies, right? All right. Then in the end, you end up with one from mom. We're doing mom. So 1, 2, 3, 4 cells with only one copy of every gene in each cell. Only one chromosome. Only 23 chromosomes. Only half the chromosome number. Because how did we get 4 cells? Because we divided the cells twice. We mixed them up in the first set of divisions. In the second set, we have the DNA.
I thought one was dominant and recessive. I thought one was heterozygous and one is homozygous. Because there's two dominant A's.
There are 2. Why are there two big A's here and 2 little A's here? Because we copied the big A's. All right, so this is just for mom. You're right that this was heterozygous. That's why you have both big A and little a eggs. All right, we'll finish this after lunch. So let me repeat that because I had forgotten to push record here. Somebody asked a really good question. I can understand their confusion. I don't understand. They said, when you have, how did they get big A, two big A's, if it was a heterozygote? Well, remember, this parent went through, took that cell in their ovary, and they did meiosis, which meant they doubled the DNA in the first part of the cell cycle, in S phase, before they went into meiosis 1. Then they did the crossing over because the tetrad formed. So that mixed up the genes. Then they separated the two homologs into two cells. Then they did meiosis again and separated the chromosomes into one chromosome in each of the new cells. But of course, they were copies of one another. So you're going to have two big A's, but there's only one big A in each cell. See, this cell has one big A. This cell has one big A. This cell has one little a. This cell has one little a. Why are there 4 copies? Because you made copies during S phase so that you can separate them. So then that gives you this combination. Now, when you did the same thing, if you did the same thing, I'm going to do you one even better. I'm just going to go ahead and just copy it. Oh, I shouldn't do that, I guess. Hold on a second. I'm just going to go ahead and just copy that. Just literally copy and paste it over here. Except in the beginning, instead of having 2 big A's, right? You had 2 little a's because it's homozygous recessive. That's this parent here, this father. When they're making their sperm, they don't have any big A's. So look, I'm getting rid of the big A's. Why am I getting rid of big A's? Because they didn't have any big A's. They only had little a's. They were homozygous recessive. They made copies. They did crossing over. They exchanged the DNA. But all the copies were little a. So they did everything that the other partner did. It's just that all the genes, all the copies of the genes were the same because they were homozygous. So they're all little a's. So all 4 sperm are little a's. 2 of eggs have big A, and 2 of the eggs have little a. Well, how can we? It seems like this is there's a lot here. There's a lot of drawing, a lot of opportunity to make mistakes. Well, this guy named Punnett came up with a way to take this and simplify it. He said, look, you have 2 options here. You start off with 2 options. Either the the mother is going to give a big A, or she's going to give a little a. That's all she has, right? Yes? And dad is going to give a little a or a little a. That's all he has, correct? By the way, let me go ahead and add to the vocabulary word. Again, all these are on the board and have been. Punnett, square. And again, we're talking about a mono hybrid. Cross, meaning one gene mixing up 2 alleles. So then the question comes in, instead of doing all this, what we just drew out, and wondering which egg and which sperm is going to go with which egg, and what are the possibilities of having a child with little a, or little a, big A, or what have you, we can just do a Punnett square and say, well, the big A here, if the mother gives this big A and dad gives this little a, that's a possibility. Mom could give a little a and dad could give a little a. That's another possibility. Then mom and dad could give that, and mom and dad could give that. So that ultimately what I end up with is 2 out of 4 have a possibility or the probability of 2 out of 4 being little a. Of course, by now you should know that 2 out of 4 is the same as 1/2, which is the same as 50%. So the other possibility is that you have a big A, little a. That's another possibility. There is no way to get a big A. If you think about it, you can see that there's no big A that this one could give. And if you think about it, it makes intuitive sense. If mom is little a, or dad is little a, how could he possibly give a big A? It's impossible. He doesn't have it to give. So it's on the only other possibility is big A, little a. And there you can see it's 2 out of 4. Again, 2 out of 4 is big A, little a. And that means, again, 1/2, big A, little a, which is, again, 50%, big A, little a. So the probability of getting, again, this is what? Is this genotype or phenotype? Genotype. I've not even mentioned a phenotype yet. Genotype, ratio. The genotypic ratio for big A, little a, and little a is going to be, bless you, 1 to 1. For every 1 big A, little a, I have 1 little a. That's my ratio. Now, if I wanted to talk about phenotype, I'd have to know, what does big A get me? Maybe big A equals tall in plants, and little a equals short. Why do I have only one letter for tall? Because it's the dominant. And what does that mean to you?
Even if it shows up once,
That's right. Even if there's only just one copy there, it has the dominant trait. But for the recessive, I need both the little ones, so that's why I put little a, so it's short. So then if I wanted a phenotypic ratio, what is this phenotypic ratio that I have here? That's right. The phenotypic ratio would be how many tall for every short. And what is it? For every one tall, I have how many short? 1. If I really just wanted to just use plain numbers, I need 2 tall for every 2 short. Well, isn't 2 to 2 reducible to 1 to 1? Okay, so that's how we get it. So that's the probability we have here of getting this phenotype or this genotype, 50-50. And it's also the ratios are here. So Punnett squares are very useful in monohybrid crosses. But guess what? It's not just, we don't have just one gene. We could use it for 2 genes or 3 genes or 4 genes or 10 genes. We'd probably use computers for anything over 3. You'll never see a problem that has more than 2. So let's keep, at least not in high school, and probably not in the college. So if you have 2 chromosomes and you're dealing with, let's say, an A, and on the other chromosome you have a B. Let's use a different color. How many copies of each chromosome do you have? Let's say this is chromosome number 2 and this is chromosome number 12. How many copies of chromosome number 2 do you have? You only have 1 copy of every chromosome? How many copies of every chromosome do you have? 2. Where'd they come from? Mom and Dad. Thank you. I meant that. I know you look like I wasn't trying to be smart. Thank you. That's exactly right. So chromosome number 2B, that maybe dad got one big B from his mom, and maybe from his dad he got a little b. So chromosome number 2 has 2 copies. They don't have to have the same allele, but they have to have the same genes. They're homologs. They're both chromosome number 12, and those 2 are both chromosome number 2. So maybe this person has this combination of genes. So how would you write that combination of genes? In just letters, rather than having to draw it all out like this. What would you write? What's the genotype of this individual? Could someone look on the board and tell me that? What is the genotype of that individual? You should be able to easily tell me. Let me zoom in case you have trouble seeing it. What is the genotype of this individual? Stop looking at your fingers and your pencil and your toenails or whatever you're looking at, and look on the board and focus and tell me what is the genotype of this individual. What is it? Big A, big B, is that all there is? That's all you see there? What do you see? Yeah.
Is it big A, little a, and then capital B, little b?
That's right. This individual is big A, big B, little b. Two copies of every gene. This is the type of genes they have. Does that not make sense? Okay. So if I'm going to, let's say that this person or tree or whatever it is matches up with another creature that has the same genotype. They're going to have babies. We're going to cross them. And we want to know how many, let's say that the A is tall, little a equals short. Let's say big B blank is blue, and let's say little b is white. Let's say that. Then you wonder, how many tall blue plants, let's say they're plants, how many tall blue plants am I going to have? How many short white ones? How many tall white ones? Now, I could sit there and stare at this for days and come up with some guessing numbers. I could do that. Or I could do a Punnett square. I'd have to remember, though, that in meiosis, I'm going to copy this chromosome. I'm going to copy this chromosome. These 2 chromosomes are going to form a tetrad. They're going to mix up their DNA. These 2 are going to do the same thing. Then I'm going to end up with 4 cells with all kinds of combinations of genes. I could work all that out. I could sit here for the rest of the day and draw it out for you, one step at a time. It really isn't that hard. It just takes a long time. There's a really short way of doing it. Again, Mr. Punnett came up with it. Punnett square. But I got a problem. I got 2 genes. Now it's going to get, this is where everybody gets confused. Everybody. I'm going to tell you right now, when I was in high school, I was confused about this. I didn't figure this out till I got to college. No reason for it. I was just lazy. It's not that hard. I was just lazy.
Is it like 8?
Well, the thing is that for each parent, this is, remember, I'm going to remind you that parent 1 goes here along this top, and parent 2 goes up and down here on this side. Each parent's going to form 4 eggs or 4 sperm for every cell. Every cell starts off with this combination. But then they're going to duplicate their DNA. They're going to mix them up. And then they're going to put one chromosome number 1 and one chromosome number 12 into each egg and each sperm. And how many eggs and sperm are they going to have at the end of meiosis? 4. 1, 2, 3, 4. And the same thing here. 1, 2, 3. Male and female, they're all going to make 4 sperm and 4 eggs because they're all doing meiosis. They're all making gametes. They're all making sex cells. Cells that they're going to use to recombine their DNA, which is what we mean in biology when we say sex. So that was problem number 1 that everybody gets confused with. They forget that that's why we have 4 on the top and 4 down the side. The next question is, well, what combination would you have? Here's what people do. Watch what people are going to do. Some of you are going to do it. Guaranteed. As soon as I stop talking, tell you to start doing your practice problems, you're just going to do it. You're going to do it in your practice problems. Some of you are going to copy those answers and submit them. You'll get partial credit. Then on the test, you're going to make this mistake because you haven't really worked it out. That's what's going to happen to some people. But let's go ahead and do it and show you the mistake that I'm predicting somebody's going to do. Somebody's going to do this. They're going to put 2 genes in every egg. I did that. I put 2 letters in every egg. Is that right?
No.
What's wrong?
Each of them has their own copy of one gene.
That's right. Each of them has a copy of each gene. One single copy of each gene. In every one of those spaces, there should be 1 A and 1 B. There shouldn't be 2 A's and 2 B's anywhere up there. 1 A and 1 B. 1 chromosome number 2 and 1 chromosome number 12. That's you only have half the DNA. Is that right, Jazz? Mm-hmm.
Why are you just calling on me?
Because I'm looking back there and you're talking to people next to you. Or you're giggling or talking or looking or whatever it is you want to say.
Okay.
All right, I'm just saying. You asked why. I'm telling you.
Okay. You can say one thing.
All right, that's okay. I'm not upset. You asked me. I told you. Why are you getting upset? All right, so how do we figure out what combination of genes are going in each of these cells? Well, we're going to do something that all of you have done forever. At least I think you have. That thing called FOIL. You've heard of FOIL. You've done it in middle school, some of you. Some of you have certainly done it in I can promise you've done it in Miss Simpson's class or Mr. Pennypacker's class. I guarantee it. And if not, if you say no, I'll call him right now, tell him to come down here.
Foil? Yeah.
FOIL, that's right. First, when you're factoring, you do first. First just means take the big A and the big B. This is just a way to keep track of them. That's all FOIL is keeping track of your factors. 1 big A and 1 big B. Then what's the next one? First and outer. That would be a big A with what? A little b. Then the inner, which is little a with big B. And then last, that's right, so some of you do remember, is last, which is little a, little b. FOIL, first, outer, inner, last. Just do that. It's just a way of keeping track so you don't make a mistake and you don't end up with 2 of these or 2 of those when you're not supposed to have 2 of those or 2 of these. Is that understood?
Yeah.
It's just that easy. So people that don't have their eyes on the board, I'm asking why not, and I know the answer, but I don't want to say their names because I don't want them to get upset. And again. All right, so we're going to, since both parents have the same genotype, they're both going to make the same kinds of gametes. So I can do this with pretty good confidence that that's what I'm going to end up with. It's just copying what I put on top. So when that egg meets this sperm, what I'm suggesting to you, some people ask me, does it really matter? I'm telling you it's the best way to do it because then you don't going to make any mistakes. You can read it easily. And if you do make a mistake, you can identify it quickly. So what you should do is put in what the egg and the sperm combo is with the letters together, the two A's together, two B's together, and start by writing the capital first and then the lowercase. So big A, big A, big B, big B, big A, big A, big B, little b, big A, little a, big B, big B, big A, little a, big B, little b. Now go ahead and finish the rest of this chart. I'm going to pause it here, and then I'll come in and fill in the chart. And you check and see if you get it right, if you got it right. And let me go ahead and fill out the rest real quick. Now that you've had a head start, let's see who gets done quickest. I think, and of course, most correct, since you can get it done quick and it be wrong.
I got the little B in the first one.
Yes, I did. You two need to stop talking to each other. In fact, there should only be one person back there, and there shouldn't be any ladies back there. Then when I make the phone call home and mom asks how come my son is or my daughter is messing up, I'm going to say, well, they were with a girl and with a boy in class. They wouldn't listen and sit where they're supposed to sit. And then you're just talking, just letting everyone know, including your mother, who we're talking about, aren't we? All you had to do is just do as I said, which is separate. Just separate. That's it. Stop messing around before I have to write a referral. That's all. That's it. Not a big deal. Go back to your assigned seats. There it is. Not that hard. I don't think I made one mistake. I left out a little B. Did we get it right?
Yeah.
All right. So that's how you're going to do it. It's not that hard as long as you do what... Say that again. All right. So when we look at now we have to look at genotypic and phenotypic ratios. Let's go ahead and take and use the phenotypic ratios first. So the first question, and what are the different combinations I could have? I can have, that's right, go ahead. Tall and blue. And let's be clear, because some people don't know this, and they're not going to say it, but they don't know it. We're all embarrassed when we don't understand something, so I understand that. But this is a phenotype, right? Tall and blue is one possibility. What's another possibility?
You said blue, white, and short tall, right?
So I can have short and blue. Right? I can have, okay, so let me do something else because I know what's going to happen. So let me do this. I can have tall and white, correct?
Yes.
I can have short and blue. And I can have what?
Short and white.
Short and white. Those are the only possibilities, right? Correct? So let's count how many tall and white ones. I'm going to put little circles, little white circles for tall and white. I'll put a nice long white line for tall and white. This one's tall and white. Do you agree? This first one here is tall and white. This one's tall and white. Notice their genotype is different. Notice that the second one has a little B, but that's okay. It has a big B. So oh, wait, that's not tall and white. What am I doing? It's tall and blue. I'm doing tall and blue, not tall and white. My bad. Let me get rid of that. Let me get rid of this and put a blue line. So tall and blue. This is tall and blue. I'm just going to have to use your name. Jazz, why are you not writing? Why don't you have, I don't understand. You know, why don't we have a conference with your folks and figure that out together? Because this is outrageous. All right, so tall and blue, even though there's a little B there. And here's a little A, but it's still tall and blue. Over here you see it's tall and blue, even though there's a little A and a little B there. This is tall and blue. The next one, is it tall and blue? No, that's tall and white. We don't want that one. But this one's tall and blue. And the next one is tall and white again, so that's not it. This one down here is tall and blue. This one is tall and blue. Next one's short and blue, which is not what we want. Next one's short and blue, which is not what we want. This one here is tall and blue. Next one's tall and white. Next one's short and blue. And next one's short and white. So that's it. Well, how many tall and blues do you count? I can tell you already. I don't need to count because I know, because I memorized this ratio, and you should too. How many tall and blues do I have? 9. If I got anything other than 9, I'd be worried. The answer is 9. I'm always when a dihybrid cross, hear me clearly so you don't make this mistake. When I have a dihybrid cross, that means 2 genes, and both parents are heterozygote. Parent 1 and parent 2 are both heterozygote. You need to separate now. Parents 1 and 2 are both heterozygote. Then I expect to get 9 dominant, where both traits are dominant. 9 individuals where both traits are dominant once you make the cross. Then I'm going to expect, well, let's count. How many of these are going to be tall and white? So I'm going to draw a little line, one white line for tall and white. So that's tall and white. This one's tall and white. This one's tall and white. That's all I have. I expect to see 3 tall and white ones. 3 tall and white. How many do I expect short and blue? I'm going to put a little dot for short and blue. Little a, big B. Oop, that's blue and it's short. Little a, that's short, but then I got a big B, that's blue. A, that's short, but then big B, that's blue. So I expect 3 that are short and blue. 3 that are short and blue. And lastly, I expect 1 that's short and white. So I expect 9 to 3 to 3 to 1. I always expect 9 to 3 to 3 to 1 when I have a dihybrid cross, 2 genes, and they're both heterozygous for both parents. Now that's a phenotypic ratio. Now, a genotypic ratio is going to be a lot more complicated. You do not have to memorize this. I'm just doing it so you can see it and see the difference. How many big A, big B's do I have? I have 1 of them. How many big A, big B, little B's do I have? I have 1, 2. I think that's it. 2 of those. How many big A, little B do I have? Big A, little B. I have 1. Do you agree with that? Does anybody see any others? I see 1 of those. How many big A, big B, little B's do I have? Well, there's 1 here. There's 1 here. That's 2. There's 3. And there's 4. So I have 4 of those. That's big A, big B, little b. And I can keep going. I just count them out. And I can say 1 to 2 to 1 to 4 to 2 to 1. And I could go ahead and list it. That doesn't do me a lot of good. If I want to know genotypic percentages, what are the odds that I'm going to have big A, big B, little b, then I just do a Punnett square and I count. That's what I would do. I wouldn't memorize this ratio. I don't think I don't think anybody will ask you that, but I wouldn't do this. I wouldn't try to memorize this ratio. But that's the difference between genotypic and phenotypic ratio. That's a dihybrid cross. That's what we call That's what Mendel did. Now, what were the laws based on all these crosses, all these numbers? What did Mendel notice that were laws? And this is Mendel. This is the guy back in the 1800s, didn't know anything about DNA, just was working with peas. He came up with 3 laws. The first law of dominance. He used the word factor because he didn't know anything about chromosomes. He says one factor, and for our sake, one allele. I'm going to use the word allele. He didn't use that word. One allele is dominant, and one allele is recessive. That's what he called the law of dominance. Now, what they mean by law is that he never saw an exception. Every time he worked with peas, he always saw that one trait was dominant over another trait. One trait was dominant, the other was recessive. That's all he saw. Today, we know we have look at this, I'm writing non-Mendel, non-Mendelian, meaning not Mendel. We know we have some other exceptions. There are multiple alleles. We know that not always do you just have 2 alleles. We know ABO blood group is a good example. And I'm going to put example, i.e., ABO blood group. We covered it. We talked about it. I drew it out. I drew it for you. We know another example of non-Mendelian genetics, and you're using your computer instead of looking at the screen and taking notes. Just some added information. I'm going to respond to your parent when we discuss it, because I'm sure it's going to come up. I'm just letting you know. I don't want any surprises. That's all. I'm just communicating with you. Not upset. Just letting you know. So multiple alleles. The other thing we know is that some traits have multiple genes. We know skin color in humans and eye color in humans is controlled by more than one gene. We've talked about that. We know that not always is it dominant and recessive. Yes, I saw it. You may go. Sometimes you have codominance. More vocabulary words. By the way, all these words are on the board, have been since we covered it a week or 2 ago. Codominant. We also have incomplete dominance. So those are examples of exceptions to Mendel's laws. Those are exceptions to Mendel's laws. Now, there's another law. He says the law of segregation. The law of segregation is when you only have one gene, and all it says is that the gene is going to segregate to the gamete, the single cells, independent. They're just going to segregate randomly. They're going to separate randomly. Now, that's still accurate. Random distribution of chromosomes. You don't know which egg is going to get the big A or the little A. Just don't know. You don't know which sperm is going to get the big A or little A. It's random. That part, as far as we know, is still accurate. However, the law of independent assortment. What that means is when you have 2 genes, like the one we just talked about, A and B, on 2 different chromosomes, he argued they would be distributed randomly, which is pretty much true as long as they're not linked. The exception to this law is when you have linked genes. Linked genes are genes that are found on the same chromosome, very close to one another. Then they're not going to sort randomly. They're always going to move together. So this Punnett square only works for genes that are random, independently assorted randomly. Those are Mendel's laws. That should allow you to finish this packet by Monday. And then we're going to start looking at how this is impacted by natural selection. And we're going to start going over some OST practice problems. Let me be very clear. If you don't do this, you don't understand what's in this video. You don't go over and practice these vocabulary words. Next week, you will be lost. It will happen. When you tell your parents, I'm not ready, I'm confused, this is hard, the first words out of my mouth are going to be, let me see the index cards, where you took these vocabulary words that we've been working on and wrote down the definitions and practiced them. Let me see the practice problems completed. Let me see that the time you checked in with me and went to tutoring and got the answers you needed. Yeah, I don't understand why you pack. You're constantly packing up early.
Who? Not me. I don't be packing up early.
You literally just packed up 20 minutes early.
That was one time I thought it was time to go.
You've done that multiple times.
I haven't.
Well, you have. Maybe you don't realize it, but you have. So independent assortment. So the law of independent assortment. Again, just says, look, the genes are going to independently assort into each gamete randomly. But we know, again, just to be clear, we talked about it. We saw some examples in X-linked trait. We saw when the genes are very close together on the same chromosome, they're going to move together. So there are exceptions. Now, does anybody have any questions about any of this? All right, so you can go ahead and do this. Finish it right now in class. I'm giving you 20 minutes. Should be easy 20-minute work. If 10 of you get together, flip quarter 10 times, you're done with the flipping. Go. Get it done. Oh, let me see the energy of the youth. Go to the office.
Now if you use